Hello students, the lecture for today will be substrate level phosphorylation. Let's remind before that how many places of biological oxidation we have. There are two places of biological oxidation. The first one is oxidation at substrate level or substrate level phosphorylation, and the second one is biological oxidation, which means energy production in the respiratory chain. Or with simple words, two places of energy production we have in our organism. Approximately 80% of energy in the respiratory chain is produced, and all the rest at substrate level. What does it mean for substrate level phosphorylation? For students, we're talking about the glycolysis process which occurs in the cytosol, and thanks to some single enzymes called glyceraldehyde triphosphate dehydrogenase and enolase reaction, there are two reactions, are produced ATP at substrate level. The second example are oxidative decarboxylation of α-keto acids with two examples of PDH complex, which is called full name pyruvate dehydrogenase complex, α-ketoglutarate dehydrogenase complex, and also there is one more complex for the branch chain α-keto acids. Let's start from glycolysis reactions. The first reaction in glycolysis is called oxidation and phosphorylation of glyceraldehyde triphosphate. Let's see, students, the substrate of the reaction. When you need to explain the reactions, one biochemical reaction, you need to start from the substrate. Explain what is this, what is the product, what is the enzyme name, or what is the cofactor name, if the reaction is reversible or not, something like that. And let's start with the substrate. The substrate of the reaction is called glyceraldehyde triphosphate. The product is 1,3-bisphosphoglycerate, and the enzyme name is glyceraldehyde triphosphate dehydrogenase enzyme, which uses NAD as cofactor, and inorganic phosphate that is used together to form one product, which is high energy compound. Why? Because here there is high energy bond. That's why, thanks to these bonds, there is possible formation of ATP at substrate level. Why the name is substrate level? Because the substrate, high energy substrate, is responsible for the formation of ATP immediately after that in the next reaction. Let's remind one more rule when we talk about energy production. Students, you know that the only reactions that can be used for the energy production are oxidoreductive reactions, which means we need to have before that oxidoreductive process to form high energy bond like this one, and that's why it's possible to form ATP at substrate level after that. Actually, these macroergic bonds together with the phosphate atom is attached to ADP to form ATP at substrate level. And the last product from the enzyme phosphoglycerate kinase is 3-phosphoglycerate. Let's see the molecular mechanism of this reaction. We start from number one, just follow the arrows and you will understand the reactions. Let's start from number one. This is the substrate of the reaction, glyceraldehyde triphosphate, which is attached to the enzyme called glyceraldehyde triphosphate dehydrogenase, which uses NAD as a cofactor. And in the active site of the enzyme, there is one cysteine residue with SH group inside, which is very important. After this attachment is formed enzyme-substrate complex, and after that there is, thanks to this alkyl group of the cysteine, after that in the reaction 2 there is dehydrogenation of the aldehyde group, and because of the oxidoreduction process is created high energy bond, which is called tyrol ester bond. And now the NAD, because takes two hydrogen atoms, is converted to its reduced form called NADH. After that, in the reaction 3, this NADH is replaced by one new external NAD which enters the reaction. That's why in this reaction, look here students, there is releasing of one NADH. The reason is because of reaction number 3 in this scheme. And after that, there is reaction number 4, which is phosphorylation. It's very interesting, by the way, enzyme which has two domains. The first domain is domain for the dehydrogenation of the reaction, and the second domain is called kinase domain because of the phosphorylation. At the same, immediately after that, there is phosphorylation, but this phosphorylation is catalyzed by the same enzyme, and the high energy compound is ready with high energy acyl phosphate bond. That's why immediately after that, in the reaction 5, there is phosphorylation of ADP, and ATP is formed at substrate level. The last compound is 3-phosphoglycerate. All these reactions are from glycolysis process and occur in the cytosol. All glycolysis occurs in the cytosol. This is the explanation. The second reaction is called enolase reaction, but also there is one more pyruvate kinase reaction. Let's see the enolase reaction, students. The substrate is 2-phosphoglycerate. By dehydration, releasing of one molecule of water and takes to the enzyme enolase, it is produced one very high energy compound with high energy bond called NO phosphate bond. The compound name is phosphoenolpyruvate. Thanks to this high energy bond formation, in the next reaction immediately after that, there is formation of ATP at substrate level. Why? Because of the high energy substrate before that, that's why. Actually, it's not a typical oxidoreductive process. It's not oxidoreduction because no NAD, no FAD, but there is so-called intramolecular oxidoreduction because the releasing of this hydrogen atom and this hydroxyl group to form water in the end. And the last product is pyruvate. This reaction of pyruvate kinase is absolutely irreversible because after this intermediate, which is called enolpyruvate, the next reaction is absolutely exergonic and leads to the end product, pyruvate. Both of the reactions are part of the glycolysis tube, which means they occur in the cytosol. What is the significance of all these reactions with substrate level phosphorylation? Number one, because under oxygen deficiency or anaerobic conditions, which means deficiency of oxygen, we need to rely on only the glycolysis process, because if there is no oxygen, the mitochondria don't work. They don't work because they need oxygen for their action. That's why no mitochondria, only cytosol. In the cytosol, there are only these two reactions from glycolysis. Number two, it is independent of mitochondrial respiratory chain, which means they are not affected by inhibitors of oxidative phosphorylation, like inhibitors of electron transport, like uncoupling agents, like oligomycin. They are not affected. And number three, when there are mitochondrial diseases, we rely on exactly these substrate level phosphorylations in the glycolysis process in the cytosol. And one more, all this ATP, which is produced during this substrate level phosphorylation, is used for the biosynthetic processes, because mainly they occur in the cytosol, students. The second part of these processes of substrate level is our oxidative decarboxylation of α-keto acid. Let's see the first α-keto acid. Some information before that about the complexes. All these processes are catalyzed by multi-enzyme complexes, not by single enzymes like the previous ones. The multi-enzyme complexes consist of three enzymes, and they use 5 cofactors. And they have very high advantages for the using of multi-enzyme complexes. Number one is high speed of reaction, reduction of side reactions with intermediate metabolites, and common regulation of all complex, not single regulation one by one. It's very important for the velocity of all process. And the examples. The first one is the PDH complex, which catalyzes the reaction from pyruvate to acetylcoenzyme A. What is the role? Connects glycolysis to the Krebs cycle. The second reaction is from α-ketoglutarate to succinylcoenzyme A, which is a part of Krebs cycle. The enzymes are the same, like these ones, but only difference is in the first enzyme. You have to know the enzyme names. Enzyme number 1 is called pyruvate dehydrogenase, which uses cofactor thymine pyrophosphate. The second enzyme is called dehydrolipoyl transacetylase, which uses lipoic acid or lipoamide plus coenzyme A. The third enzyme is called dehydrolipoyl dehydrogenase, which uses 2 cofactors, NAD and FAD. And the only difference is the enzyme number 1 for the next complex. Let's start and talking about pyruvate dehydrogenase complex. This is the first enzyme, the second one, and the third one. The first cofactor that we need is thymine pyrophosphate, which comes from vitamin B1 or thymine. It's a derivative of B1, and actually there are 2 rings. Contains the pyrimidine ring, this one, this is the thymine pyrophosphate, and thiazole ring. The thiazole ring is key to the binding and decarboxylation of pyruvate. This is the first reaction. This is the second enzyme, which uses 2 cofactors, and lipoic acid and coenzyme A. And let's remind the coenzyme A structure. There is pantothenic acid in the structure of coenzyme A, which was vitamin B5. In the end of the structure, there is one cysteamine with SH group. The third enzyme uses as a prosthetic group FAD, which converts to FADH, and after that back to FAD by using NAD, which is the second cofactor for the third enzyme. And let's go directly to the scheme, students, to explain the scheme of oxidative decarboxylation of pyruvate to acetylcoenzyme A. This is the substrate of the reaction, pyruvate. This is the product of the reaction, acetylcoenzyme A. Let's see the cofactors. Cofactor number 1, this one, PPP. Cofactor number 2, lipoic acid or lipoamide. Cofactor number 3, coenzyme A. Number 4, FAD, and number 5, NAD, 5 cofactors, and 3 enzymes. Enzyme number 1, I said the name. Enzyme number 2, I said the name. And enzyme number 3, I said the name too. And just follow the arrows to understand the process. Let's start from the first reaction, which is catalyzed by the first enzyme, which works together with TPP as a cofactor. The reaction is decarboxylation. This blue carbon dioxide is released and hydroxyethyl group is produced, which is together with TPP. After that, the reaction number 4 is catalyzed by the second enzyme, which uses the first cofactor, which is lipoic acid but full oxidized form. Actually, there are 3 forms of lipoic acid: the full oxidized form, the full reduced form, and the semi-reduced form. The first oxidized form accepts this acetyl group, but together with dehydrogenation, and the disulfide bridge is opened. And now the acetyl group is connected with the lipoic acid. Then the same acetyl group is transferred and accepted by coenzyme A to form acetylcoenzyme A end product. Actually, we are ready. But for the regeneration of lipoic acid, we need to continue the reactions. Reduced form of lipoic acid is produced with 2 SH groups. Then, reaction number 4. These hydrogen atoms are accepted by FAD and the lipoic acid is full oxidized. But these hydrogen atoms are in the form of the reduced form of FAD called FADH. Then the same hydrogen atoms, thanks to the third enzyme, are accepted by NAD to form NADH, which goes directly to the first complex of the respiratory chain, and 3 ATP molecules will be produced in the end. That's all the mechanism. Now let's talking about the products and the substrate of the reaction, because to clarify the allosteric regulation after that. To understand the allosteric regulation, students, you need to know one very important rule. The main inhibitors of the reaction are the products. The main activators of the reaction are the substrates. That's why we need to clarify which are the products. Let's see. I said that the final product is acetyl coenzyme A. This is the product. But because there is participation of many cofactors, they can be produced many so-called co-products from some cofactors. And this one, which is plays a role of product too, is NADH. NADH. Why not lipoic acid? Why not lipoic acid to be accepted like product? It's not a product because lipoic acid is regenerated in this cycle. Is it can be possible to be TPP a final product? Of course, the answer is not, because it's regenerated in this cycle too. Is it possible to be FAD? Again, no, because FAD is regenerated in this cycle, but only one that is produced and goes out of the mechanism is NADH. That's why acetyl coenzyme A and NADH are the final products, which means these are the allosteric inhibitors. Substrates. The substrate of the reaction is pyruvate, but also there are 2 cofactors that just enter without regeneration. Which are they, students? Can you see from the scheme? These are NADH and coenzyme A. Only these ones just enter the reaction. That's why these are allosteric activators for the process. One more regulation there is, except allosteric, is reversible covalent modification. This complex entirely can be regulated by reversible covalent modification, and the active form is PDH with dephosphorylated form. This is the active form. This is the substrate of the reaction, pyruvate. Here is written product of the reaction, acetyl coenzyme A. This is the inactive form. You know this scheme. Pyruvate kinase catalyzes the phosphorylation and inhibition. Protein phosphatase catalyzes the dephosphorylation and activation. Because we know that insulin activates phosphatases, this means insulin activates the PDH complex and oxidative decarboxylation of pyruvate. The differences with the regulation of α-keto-glutarate dehydrogenase complex is only this, that there is no phosphorylation, dephosphorylation like in the PDH complex. About this one, no. There is only allosteric regulation. And one clinical case. Okay, let's see the medical history of one disease, which is called pyruvate dehydrogenase deficiency. The patient is a male child born at term as a result of a normal pregnancy and delivery. 2 months after birth, he was admitted to inpatient care and he stopped gaining weight and appeared lethargic. From physical examination and status, upon admission, the child is in a bad general condition. He has reduced muscle tone, hypertension, and deep, rapid breathing. His body weight is below the norm for the corresponding age. From the laboratory results, we can see that the lactate quantity is very, very high. The pyruvate quantity is very, very high. The alanine too. The pH is very low. It is called acidosis, but the glucose level is normal. The result, high pyruvate, high lactate, which is called lactic acidosis because of the low blood pH, high alanine, and let's go directly to the scheme. Why the alanine, lactate, and pyruvate are so high? Why? Actually, students, this is the scheme. Pyruvate is produced from glucose only. This is the process. Not only can be produced from alanine too in this reaction, the opposite reaction from alanine, but mainly from glucose. One more, pyruvate can be produced also from lactate by this reaction, the opposite reaction, this one and this one. If there is deficiency somewhere in this complex, PDH complex, no action of this enzyme, what will be the consequences? Actually, let's remind the consequences of enzymopathies. What was this enzymopathy? Enzymopathy means deficiency of some enzyme because of the mutation in DNA. Can be reduced or full lack of the enzyme. And if we know that the lactate is very high, the alanine is very high, and pyruvate is very high, which means that maybe the reason is glycolysis, but actually no, because the glucose was normal, because no deficiency in the enzymes from glycolysis or gluconeogenesis. But if there is a deficiency of this enzyme, the pyruvate will be increased, and all these derivatives that can be produced from pyruvate. And the causes of this PDH complex decreased activity can be the cofactors. Why not? There are 5 cofactors, but can be different genetic defects in these 3 enzymes of the PDH complex, and also one more enzyme which is important for PDH complex, which is phosphatase. Why phosphatase is so important? Because it is responsible for the phosphorylation of PDH complex to become dephosphorylated form, which is active. That's why it's very important, this enzyme. And the common defects in E1 are about the 2 subunits, alpha and beta subunit, and alpha subunit play a key role for the contact with serine residue because of the content of one hydroxyl group of the serine, which can be phosphorylated and become active. After the mechanism actually is point mutation, which impair the binding of TPP to PDH phosphatase, and after that the complex remain inactive phosphorylated form. This is the mutation, and this is the mechanism of this mutation. Additional investigations can be if there is high dose time in supplementation, but there is only temporary decrease in the plasma lactate. PDH activity assay in skin fibroblasts can be tested, and there is low basal activity and very low affinity for TPP and PDH phosphatase. The conclusion is confirmation of a defect in thymine pyrophosphate and PDH phosphatase binding. This is the problem of this complex. And the therapeutic approach, how can be? Actually, the therapeutic guidelines are standard thymine treatment is ineffective in the defect lies in TPP binding to the enzyme, And because the main problem is pyruvate is primarily oxidized to lactate, which means lactic acidosis, increasing the lactate. The dietary approach would be limit carbohydrates. Why? To decrease the formation of pyruvate, because the main process for the formation of pyruvate is glucose. That's why we need to reduce the carbohydrate food in these patients. Number two, to prefer lipids as a main source of energy, which means lipolysis, fatty acids and glycerol. Then fatty acids will go to the beta oxidation, and acetyl coenzyme can be produced, but not from pyruvate. From beta oxidation will be. The main goal is to decrease the plasma concentration of lactate and alanine. And that's all from today's students. Thank you for your presence.